Hey there, welcome to my video. My name is He Qian. I'm an NR Fellow and Assistant Professor in the Department of MSE. My research mainly focused on developing and applying electron microscopy techniques to understand nanomaterials, and I'm particularly interested in catalysts. Some of the images of nanoparticles, nanoclusters, or even isolated atoms are shown here, and these materials can be the key to solve many of the grand challenges we're facing today, such as sustainability, environmental protection, and food security. Today, I'm mainly going to introduce the electron microscope, which is a major research tool in science. As the late theoretical physicist Freeman Dyson famously said, the great advances in science usually result from new tools rather than from new doctrines, and this is especially true for microscopy. If you scan through the history of science, every development of microscopy techniques will always lead to new discoveries. For instance, it was the visible light microscopes that opened up the world of micro. Here is the first picture of cells observed by Robert Hooke using his microscope back in 1665. And since then, there have been continuous attempts to build better microscopes to see smaller and smaller things. However, scientists soon realized that the microscope is limited by the wavelength of the light source. And in order to obtain shorter wavelengths, electrons were used. And the electron microscope was invented by Ernst Ruska in the 1930s. And it is the electron microscope that opened up the world of nano. With this new resolving power, scientists discovered dislocations in structural materials such as steel, which basically explain their mechanical properties so that we can develop lighter and stronger materials. Another famous discovery is carbon nanotubes, which were first observed by Ijima in 1991 under an electron microscope. And the evolution of microscopy did not stop there. For about 60 years since its invention, the limiting factor for electron microscope was found not to be the wavelength, but the quality of lens. Basically, the electromagnetic lens used in the microscope are not perfect and cannot focus electrons into a sharp point due to the existence of lens aberrations. Successful aberration correction was achieved in the late 1990s, and the main inventors were just awarded the prestigious Kavli Prize in Nanoscience this year. Just watch, Nobel Prize one day will also not be a surprise. And because of the aberration correction, microscopy just had another revolution of resolution, and nowadays microscopes can resolve things less than 1 angstrom apart, and that is 1 to the power of -10 meter. And we have such a microscope in NUS. It's called aberration-corrected scanning transmission electron microscope, or AC-STEM in short. It is extremely powerful in terms of resolution, and is also highly versatile. Here is a schematic showing how it works. A focused electron probe less than 1 angstrom in diameter will be rastering over a thin specimen, and different signals from electron-metal interaction can be collected to form different types of images. For instance, electrons scattered into higher angles can be used to visualize heavier atoms, such as transition metal cations, while the electrons scattered onto lower angles can be used to observe lighter atoms, such as oxygen. AC-STEM can also do chemical analysis through X-ray or electron energy spectroscopies. So we not only can know where the atoms are, but also know what types of atoms they are, as well as their other chemical formations, such as bonding configuration or oxidation states. This microscope is also highly unique. It's the only one in the US and also in the whole Singapore. This powerful microscope is an important player in many research activities in the US that are either pursuing better batteries, more efficient solar cells, or novel 2D materials for next-generation computers. But today, I want to mainly discuss the challenges we're facing so that the excellent young graduate students can help to resolve. I believe the first challenge is the so-called vacuum gap. Electron microscopes normally work in high vacuum to prevent electrons get scattered by air molecules. However, many materials, especially catalysts, can have different atomic structures under a vacuum compared to those under their actual working conditions. It is highly desired to carry out so-called in situ microscopy where the materials is sitting not a vacuum but in a realistic environment. Now we have a new opportunity to do just that. We just installed a state-of-the-art in situ gas holder. Now we can study materials at 1 bar pressure of different gas environment and up to about 1000 degrees C. This will allow us to have a new look of many industrially important materials. The experiment can be quite challenging, but the reward is also very high. The second challenge I want to introduce is the so-called electron beam damage. Basically, electron microscopes nowadays are no longer limited by lens aberrations, but by the electron-matter interaction itself. Many important materials, such as organic materials, are very prone to be altered by fast electrons, so they are very difficult to study under electron microscope. For instance, some materials can be amorphized, and some others can be recrystallized under the electron beam. To battle this challenge and to study the intrinsic material structure, we need to aim for electron dose efficient microscopy, where we try to get the most out of the limited dose determined by the damage mechanism. And the new opportunity nowadays is that there are new generations of electron detectors, where the electronic readout noise are eliminated. These detectors will also facilitate new imaging mode, such as scanning diffraction or 4D STEM, where a full diffraction pattern is recorded at each pixel, rather than just the integrated electron intensity. This will allow traditionally difficult materials to be studied in the electron microscope, and new discoveries can be expected. The final challenge I want to mention today is a high-throughput microscopy challenge. Nowadays, chemical synthesis and material testing can be highly automated, and there are even robots running a chemistry lab now. On the other hand, computational material science is also developing fast, and high-throughput material screening from supercomputers are now possible. High-throughput material characterization, especially microscopy, appears to be the missing link to complete a fast material development cycle. And obviously, we're living in the era of artificial intelligence now. From self-driving cars to beat the best Go player, AI continues to impress us. And I'm sure one day, self-driving microscopes will be possible for material science. And a lot of research effort has already been on this, from image recognition to data mining to decision making. And this is an area with a really high potential. Okay, I will stop here. We'll welcome any motivated and talented students to join us for microscopy endeavour. Just shoot me an email so we can discuss more. If not so interested, that's okay. But if you are still working in the field of material science, we also welcome you to make use of this powerful microscope, which I'm sure will help your research. Thanks for watching.